Great. Well, welcome everyone to our AMS monthly seminars. I'm Jonathan Plett. I am the president of the council. And it is my pleasure today to be welcoming Adam Frew as our invited speaker. Now, Adam has interest in a number of different areas. He's an ecologist that is looking at the impact of arbuscular mycorrhizal fungi, but in a very holistic sense. So looking at that idea of how colonization of arbuscular mycorrhiza not only affects plant health, but how it affects their interaction with insects. If you look at his website, there are three main areas that he is working in. One is that aspect that we're talking about today, that AM plant-insect interaction. He's also interested in how AM fungi are affected by disturbance and how their community assembly is impacted by that. But he also has a really cool citizen science project that he is running called Dig Up Dirt, where he is working with different farmers, landowners, and land managers and asking them to dig up dirt and send it to him so that he can survey different AM fungal communities across Australia. So if you're interested in that, please do go to digupdirt, all one word, .net for more information. So Adam did his PhD at Western Sydney at the Hawkesbury Institute for the Environment back when I started at the Institute here as well. He then proceeded on to a postdoc at Charles Sturt University, then was hired on as a lecturer at the University of Southern Queensland. But thankfully for us, he has returned to the HIE where he took up a DECRA fellowship a couple of years ago. So with that, I will pass it over to Adam today, who is going to be talking about this tripartite interaction. Adam, to you.
Thanks, Jonathan. Everyone can hear me? Cool. All right. So I just want to start by thanking Jonathan and Camille for asking me to give a talk. And also thanks to everyone who is dialing in and taking up some of your lunch break or lunchtime, I suppose it probably is for most of you. Maybe it's a bit later after for the New Zealanders, to listen to me talk. So my talk's called Shared Host Divided Interests. Shared host because I'm talking about arbuscular mycorrhizal fungi and insect herbivores, both of which their host is plants or terrestrial plants. But divided interests because I suppose primarily they're interested in themselves, their own fitness, their own success. Although I suppose they do have similar interests in the sense that both of these groups are looking for resources from their shared host plants. So they just go about it in, I suppose, very different ways. So we have this three-way interaction, this tripartite relationship, which is pretty old. And what I'm going to do is I'm going to start by giving a bit of an introduction to the oldest of these interactions. Now, of course, the oldest of these interactions is that between arbuscular mycorrhizal fungi and plants. So to do that, it might be a bit much for your lunchtime on a Wednesday, but if you can just come into my time machine and we'll just move through space and time as galaxies fly by us. And oh, we've arrived here. It's Earth, believe it or not. And it's about 500 million years ago. It's pretty boring. There's nothing happening. That's not probably quite accurate. But as you can see, it's not exactly the green planet that you're used to. I've probably gone a bit too far back, but soon the planet will see the first handshake that happens between some of the first plants and fungi. So we'll just hop back into the time machine. We'll just go a little bit forward. Here we are, and okay, this looks about right. There's a little bit more going on here. We've got some greenery. So maybe some photosynthesis is happening. Some simple plants. There's a couple of geysers there in the background. So here we have some of the first terrestrial plant life colonizing the land. But even at this point, these early plants were already associating with arbuscular mycorrhizal fungi, or arbuscular mycorrhizal-like fungi, I suppose. And so even here, beneath the soil, what we might call soil, we already have hyphae, fungal hyphae networks, depending on your definition of network, of mycelia connecting to and associating with these very simple plants. So this relationship between plants and mycorrhizal fungi is very old. It was here pretty much from the beginning of terrestrial plant life. How do we know that? Well, we know that for various reasons, but one of them is because we have some really awesome fossils. So I'm sure a lot of you have already seen some of these images because arbuscular mycorrhizal people love to show them, but this is a fossil, a cross-section of a fossil of a sporophyte of Aglyphyton major, which is about 412 million years old. And it's so well preserved that you can even see the cells of this plant. And in fact, it is so well preserved, if we go closer, we can actually see the cells, but structures within the cells. So here we're looking at a structure within a cell. And this, what we're looking at here is an arbuscular mycorrhiza. Which is a structure formed by arbuscular mycorrhizal fungi. So this is a 412, 415 million year old arbuscular mycorrhiza, which is pretty awesome. And I'm showing this to just highlight how old this relationship is, how important it's been for evolution of plant life on land. And that this is the ancestral state for all terrestrial plants that we have today. Okay, so I've talked about the origins of this relationship, but I haven't actually said much about what it is. So I'll just give you a bit more on that. So basically, to put it simply, the relationship between plants and arbuscular mycorrhizal fungi is one where the fungi will associate with the roots of plants, with the roots of plants today, and the fungi will provide the plant hosts access to resources. Now, with AM fungi, we often, I suppose, think of one of their main functions is access to phosphorus. Phosphorus is not very mobile in soil, and plants struggle to have access to that. So it's often a limiting nutrient. So the fungi will provide phosphorus, and they can also provide other things. But they don't do this out of the goodness of their little fungal hearts. They do it because they need to. AM fungi are obligate symbionts. They must have a host for their success, for their survival, because they require the carbon. So they require the photosynthase from their host. So they get the carbon, they get this as sugars and lipids from their host plant, while they provide the plant with phosphorus. I suppose that's a simplistic overview, but it gives you an idea of this sort of transfer of resources that sometimes will keep everyone happy. So often it's a mutualistic interaction, but not always. Okay, so that's a very quick summary of the arbuscular mycorrhizal and plant relationship. We haven't introduced the insects yet because they don't exist yet. We're still back in time, remember. So we just need to hop back into the time machine. Here we are, whoopee, and here we are. We've just jumped, wow, we've just jumped about 100 million years or so. This is about 350 million years ago. It's looking pretty different. First thing you notice is there's lots of greenery, there's lots of plants. There's even there's some invertebrates, animals kicking about. But at this point in time, the war that we have still today between plants and insect herbivores, so these are just insects that feed on living plant tissue in one way or another. The war has been already it's already been raging for some time. Now you might think, okay, well, there's a war between insect herbivores and these plants. Surely the insect herbivores are going to win this war in the end. Plants are sessile. They don't move really, I suppose. Insects move mostly. Insects also can eat a heck of a lot. And even if the individual insects don't, they certainly can be in high enough numbers to consume a lot of plant material. So eventually, surely the insects are going to pretty much eat all the plant life. Now, obviously, we are from the future, so we know that's not happened. We still have plants today, and that's largely because plants, to deal with insect herbivory, evolved a diverse array of defenses. So ways in which they or things they can rely on, or strategies that they have to deal with insect herbivory, one way or the other. So I'll just quickly mention some things about plant defense. When we think about plant defenses, we tend to often categorize them into two main groups. So we can think of when a plant's being attacked by an insect herbivore, plants can rely on tolerance-based defense. So essentially, this is the ability for the plant to regrow and reproduce after attack. And essentially, this is often thought of synonymously with things like with compensatory growth of the plant. So it basically just grows more to deal with loss of tissue and resources from the herbivores. So this is tolerance. Now, if you think of this in the context of AM fungi, obviously AM fungi provide resources to the plant. Key one we're thinking of often is phosphorus. So yeah, okay, the AM symbiosis is probably going to affect tolerance because a well-resourced plant is going to grow better, to put it simply. Now, another type of defense strategies are resistance-based defenses. So these are defenses that will affect the preference or the performance of the insect herbivore themselves. So these are often things like secondary metabolites or other compounds. They might, for example, reduce the digestibility of the plant tissue. They might actually be toxic to the insect itself. They might affect the fecundity of the insect, or they might affect the preferences of the insect. It might just be the toughness of the leaf, various things that will reduce the performance of the insect or the preferencing of the insect for that species, for that plant. So a lot of these mechanisms are expensive energetically. So again, AM fungi can affect these. But we also know that AM fungi do more than just provide the resources to the plant. They also are involved in very complex plant responses to herbivory, as well as the complex plant responses to colonization by the fungi. And I won't go too much into detail about that, at least for this talk. But it's just important to know that we have these defenses that AM fungi will affect. So I've talked a lot about this, but how then do AM fungi affect insect herbivores, right? Because actually there's a lot of research that's on this question and related questions, because, well, it's interesting, but it's not surprising that a lot of research has gone into this because of plant protection. So we're always looking for ways, right, to increase crop protection, to reduce damage to crops and to food production, to improve food security. So there's a lot of research that's gone on to figuring out how AM fungi can affect plant defenses and affect insect performance. So there's a couple of meta-analysis that I always tend to rely on. They're pretty old now, but they're still got a lot of value. So we know that AM fungi can have positive effect on insect herbivores. So they can actually, maybe it's not correct to say they reduce defenses. Some cases that might be the case, but they actually promote herbivore performance. They can also increase plant defenses, so they can have a negative impact on the insects, or they can have no effect. This variability is depending on a few things. So insects that are chewing insects or piercing insects, so the chewing insects tend to be more negatively affected. So they're more maybe susceptible to certain plant defenses. It also matters the location of where the insect's feeding. So Root herbivores, for example, tend to be more consistently negatively impacted by the AM symbiosis. Whether the insect is a generalist or a specialist herbivore, generalists tend to be more negatively affected by AM fungi. Host plant identity makes a difference. So the amount of benefit in terms of defense a plant gets, or from the other perspective, the amount of negative impact, the strength of the negative impact on the insect, but also AM fungal species identity. Now, on that last point, that's the point I'm going to focus in on, because even though there's been a whole swathe of research that's on this, there's so many studies, particularly within more agricultural focused research on how AM fungi can affect plant defenses. Most of this work has really focused on looking at single species of AM fungi. It tends to be the same AM fungal taxa that they work on, which is really just a handful of species. And so while there's been a lot of research, it's still very limited in terms of our understanding of how AM fungi can affect defenses. So that's looking at specific individual taxa. And even then, if we look at those that have looked at more than one species of AM fungi, there's far less studies that have involved more complex communities. So this was something that we were interested in. This is just a very simple study that we conducted comparing plant phenolic mediated resistance to insect herbivory, where we grew wheat this time, and it was wheat with no AM fungi, with a single AM fungal species, with 4 AM fungal species together. These were commercial inoculants, in fact. And then we also compared the outcome with a natural community, which was just sourced from I guess a semi-natural field, a semi-natural area adjacent to an agricultural field. So we measured phenolics. So this is a broad group of compounds. They have many functions, but they are often correlated with insect performance negatively. So they're often correlated with defense. So with 1 AM fungal species, this was Rhizophagus irregularis. With 1 AM fungal species, the phenolic concentrations in the plant weren't that different. With 4 AM fungal species, we saw a significant increase in the amount of phenolic defenses in the plant. And we saw about the same level of phenolic compound in the plant with the natural community of AM fungi. So we're seeing here differences that we don't see effects if we're working with a single species. But if we then include 3 more species, then we get an increase in those defenses, but we don't see any further benefit or any differences, at least with phenolics, from the natural community. And we also found, this is just looking at the relative growth rate of the insect, that we tended to see the lower performance, so the insect was growing less, essentially, when it was feeding on those plants that were associating with the last 2 treatments there, so with the 4 fungal species or the natural community. So I suppose the point there is that AM fungal communities can have distinct effects on plant defense outcomes. That was just one example of something that we've done. I've done other things. And as I said, there's a whole load of research that have looked at different aspects of AM fungal enhanced resistance, but focusing very much on just a handful of fungi. So for the rest of the talk, I just want to sort of switch things around. And rather than talking about how the AM symbiosis or AM fungi can affect plant defenses and insect herbivores, how does herbivory affect the AM symbiosis, and how does it affect the fungi themselves? So if we want to think about this, let's go back to the basics of what we might expect. How might we expect herbivory to affect AM fungi? A common understanding is that we would expect herbivory to weaken the symbiosis. Plants photosynthesize, they fix carbon, most of them do. And so they capture this carbon and they deliver it to the fungi, and the fungi will use this carbon, they need it to survive, and then they will deliver phosphorus to their host. Again, I know that's a very simple explanation, but it's one we work with. If we introduce an insect herbivore, it's going to eat some of the tissue. It's going to affect the plant's capacity to photosynthesize. And so we might expect a reduction in carbon, and then there's less carbon resources going to this fungal community below ground. And so if we have less carbon resources, we might expect less AM fungi and maybe less delivery of phosphorus to the host. Okay. if we look at some of the data, now this is a meta-analysis. Again, it's, I suppose, old now, but still very helpful to give you an idea of the kind of patterns we see. So this is just looking at colonizations. This is just looking at the amount of AM fungi in the roots of plants. And it's showing you the effects of herbivory. Now, you'll see most things are sort of below or lower than zero. And for a few of those different categories, the error bars don't cross. So we tend to see more often than positive, at least, we tend to see negative impacts. So we are seeing a reduction in the amount of fungi in the roots. This isn't always the case, and it does seem to matter what kind of herbivory we have. So whether it's like simulated herbivory, real herbivory, et cetera, or the type of functional plant group. Okay, but we do tend to see reduction. Whether that's biologically or ecologically meaningful is another conversation. And we've also found similar outcomes. So this is just from another experiment where we just looked at 2 native Australian grasses. We found, at least with arbuscular colonization, a reduction in the extent of colonization of arbuscular, presence of arbuscules in the roots. So we know that herbivory it's variable, but we might more often than not maybe see a negative effect of herbivory. So that's just looking at the presence of AM fungi. But what about the communities of fungi? Because AM fungi are not a monolith. They have diversity. It's a very interesting diversity. There's several hundred, if not closer to 1,000 species of AM fungi. And they're diverse, they're functionally diverse. We know this. So how do the communities respond? Because this is important. There's actually very, very little data. There's very few studies that have really examined this question of herbivory impacts on AM fungal diversity, particularly insect herbivory. There's studies that have looked at grazing. Now, of course, grazing from a large herbivore is quite different. There are other things that are impacting that, are playing into that interaction. But the studies on grazing tend to see a reduction in species richness. So we have less AM fungal species with little evidence of significant impacts on community composition. If we look at insect herbivory, again, very few studies, you could count them on one hand, found that tend to find not strong effects of species richness. Well, again, there's maybe 1 or 2 studies, but we do see impacts on the community in terms of evenness. So we're getting changes in the structure in the relative abundances of some of those taxa. So that doesn't really tell us much though, right? With so few studies, it gives us a bit of an indication. So this is something that I'm really interested in. And so of course, we did some experiments looking at it. And so we did a very simple experiment. This was, well, that's my drawing, but it was actually on sorghum, but that's obviously not sorghum. But it was on sorghum. So we basically grew sorghum with an AM fungal community and without an AM fungal community. This was a diverse community. And we basically then grew those plants with an insect herbivore and without an insect herbivore. We sequenced the roots to look at the community, the diversity, the composition. And we looked at other parameters in the plant as well to see how the plant was responding to this. So what did we see? I don't want to take too long, so I'll dive right into the results. So this is looking at the VT richness. This is basically virtual taxa richness, virtual taxon richness. Think of it just as species richness, number of species. I will say that this is actually quite a low species richness generally in the roots that I tend to find in experimental setups anyway, but that's what it is. So here, this is looking at no herbivore with the herbivore. You can see we're getting less species richness in the roots of plants with the herbivore. If we look at community structure, look at the beta diversity. So here, this is our ordination, which is based on Bray-Curtis distances. So essentially every point on this ordination represents a community from a sample. And the closer those points are together in ordination space, the more similar they are, those communities. And so you can see the width, the communities that were with the herbivore are different from those without the herbivore. So we're seeing changes, not just in richness, the number of species, but also in what the distribution of the abundances of the species and who they are. Aside from that, we also looked at the phylogenetic diversity and phylogenetic structure of the communities. So this is thinking about, suppose, the phylogeny. It's thinking about the relatedness of the fungal species that we find in the roots with and without herbivory. This is looking at Faith's phylogenetic diversity, which is a very simple metric, or it's one of the simplest metrics, I suppose. This is the standardized effect sizes. Here we saw an increase in phylogenetic diversity. So this is quite interesting. It's quite common for us to see that phylogenetic diversity takes a similar sort of pattern response to species richness. But here we're getting an increase in phylogenetic diversity. So herbivory is reducing species richness, but it's also increasing total phylogenetic diversity. Now, as well as that, we looked at the phylogenetic structure of the communities. And to do this, we looked at several things, but these are the things I'm going to tell you about. We looked at two main indices, which is the mean pairwise distance and the mean nearest taxon distance. Essentially, these tell you about the relatedness of the community, the relatedness of the individuals within the community. So mean pairwise distance is the mean relatedness between every pair of individuals within a community. The mean nearest taxon distance gives you the relatedness between the two most closely related individuals in that community. So there's slight nuances to those indices, but essentially the higher the more positive the values in this instance, the more phylogenetically over-dispersed those communities are. The more negative those values, the more phylogenetically clustered those communities are. By phylogenetic overdispersion, I mean that the individuals are less related to each other, so they're more distantly related. Clustering is they're more closely related. Now, one understanding of overdispersion, which you can clearly see that we're seeing with the herbivore, both of these metrics we're getting a higher value, so we're getting more overdispersion. So these communities are composed of taxa that are less related to each other. One way to interpret this, or our main way to interpret this, is that this is driven by competition. So with competitive exclusion. So the idea being that when you have competition, that individuals that are more closely related fulfill a very similar niche. And so when there's a competition pressure, you're more likely to lose individuals that are more closely related. And so you end up with a community composed of distantly related individuals, which is phylogenetic overdispersion. And so this is thought to be driven by competition. So is this what we're seeing here? I suppose if we think about it, we could think, yep, that makes sense. We're getting herbivory, we're getting less carbon, ostensibly less carbon going to the fungi, less resources for these fungal communities. They're going to fight over it. They're going to have competition, and we're going to lose all the related ones, and we end up with over-dispersed community. Okay, that could be a way to interpret it, for sure. But how are the plants responding to this? We'll here just focus on the mycorrhizal growth response and the mycorrhizal phosphorus response. Basically, this is telling us that the plants under herbivory, with those communities, were getting more benefit from AM fungi than they were from the other communities without the herbivore. This is comparing them to the non-mycorrhizal controls. So essentially, they were getting relatively greater growth response, greater phosphorus response. So in essence, they were getting more benefit from these communities. We also saw an increase in the frequency of arbuscules. So what does that tell us? What do we think about that? Well, I'll just go over it. So we've got herbivores, and we're getting a reduction in species richness. Okay, we're getting less AM fungal species in the roots. But at the same time, we're getting phylogenetic overdispersion. So the species within those roots are less related to each other. Okay, this is ostensibly because of competition, although it's another talk, we can go right into that, but competition. And we saw stronger positive effects of the AM fungi under this treatment. And so we saw greater plant growth benefits and phosphorus benefits. And so a way you might think of it is, okay, well, competition is happening. We get more intense competition, and the strongest wins. And in this case, it's the best nutrient provider, right? The one it's the best at providing nutrients or resources to the host. That's the one that's going to be most competitive, and so that's the one that's gonna outlast the others when resources are low. And that's why we're getting maybe a more positive response. Sure, that could be the case. But an alternative way to think of it is that if that was what we saw, if we were seeing that competition was leading to the best phosphorus deliverer, we could argue that we would expect them to be clustered, because if effective phosphorus delivery is associated with certain traits in those fungi, and so we would expect then that more closely related individuals would exhibit those similar traits. And so we would actually expect clustering of the communities rather than overdispersion, whereas we saw overdispersion. So it's still possible that could be the case, but it's probably more likely that what we saw was an outcome based on functional complementarity between these taxa. So functionally complementary taxa, which then overall has a positive impact on the plant. So, for example, this is maybe a silly way to describe it, but for example, you could have one AM fungal species that forages for phosphorus. Maybe it grows its network, its hyphae like this, and then it grows out far away from the root zone. And then you have another one which has a very different sort of foraging strategy and how it forms, and maybe it forages close to the root. But together, those two fungi with very different strategies or morphologies actually have an overall more positive effect on the plant. So there'll be many ways that could exhibit, but this is a more likely outcome, I think, considering the data and considering what we see from the communities. So good, I'm not too long, getting close to the end. So just to summarize, we have plants, we have fungi, we have insect herbivores. And we know that AM fungi can affect plant defenses against insect herbivores. There's a great diversity in this, and it matters what species or communities of AM fungi are associating with the plant. On that point, we have maybe less data. Well, we do certainly have less data compared to other aspects of that research. So we do need more information on how differences in community composition, in community structure, in diversity, in composition, being able to link certain AM fungi to certain defense outcomes. We still have a long way to go. We have very little data when it comes to those kind of questions. And then the other way around, herbivory impacting AM fungi. We know that herbivory can have a very strong effect on the community composition and the diversity of AM fungi and roots, but we still have so little data. We're quite a bit away from being able to make huge, conclusions of how this is coming about, how the real role of herbivory in the assembly and the coming together of the communities in the roots of plants. So this is something that we definitely need to have more focus on. Firstly, because it's really cool, but also because this is a really ubiquitous interaction, right? Vast majority of plants associate with AM fungi. Pretty much every plant will deal with insect herbivory in one way or another at one point in its life. So these interactions really are found across all different landscapes and ecosystems. And so understanding how they're interacting is important. Yeah. Okay. With that, I'll finish. I wasn't too long. Thank all these people and more. And thank you.
Fantastic. Thank you very much, Adam. It's time for posing questions. So many questions in my mind that have popped up. The first one is from Nicola Day from New Zealand. What do we know about AM fungi or other mycorrhizal types and other forms of herbivory, for example, mammal herbivory in that case?
In terms of defense, I suppose I certainly would not be able to speak to defenses like resistance against mammalian herbivory. In terms of the other way around, because I suppose the mechanisms would have to be quite different and at a different scale. But in terms of the other way around, in terms of mammalian herbivory on AM fungi, I think, as I mentioned, there are some studies that have looked at like grazing effects on fungal community composition, on diversity. But even then, it's really only a handful of studies, and it's maybe less empirical. Maybe it's a more observational data that they've got. Um, and so I don't actually really have an answer on that because we don't have an answer on it yet. There's definitely effects, but it's always hard to sort of disentangle with those large herbivores, the effect of actually tissue loss versus the trampling and the other like soil compaction, these other sort of things that will co-vary with the herbivory. Yeah, so I suppose that doesn't really answer your question, but
You'll have to go around and collect kangaroo spit and, put
yes,
on leaves.
yeah, well, yes. I've heard of people doing that apparently, but yes.
Yeah, they used to do that in North America with deer, I think it was deer
Mm,
saliva, and they would put it on wounded plants.
mm.
While we're waiting for more questions to come in through the Q&A, remember, please pop in your questions through Q&A. Hi Guy has one. Great talk. In the last experiment, where were the herbivory and no herbivory treatment communities nested within one another, or was there high overlap?
There was some nesting, but the herbivory community also had some taxa that were unique to that community. So there was, I suppose you could say, potentially recruitment of other taxa that weren't either present or in very low abundance without the herbivore. So yes, you can read into that what you will, but there could also, you could also think of those interactions as not just being about resource use or resource availability to the fungi, but also maybe selection by the plant for particular fungi, or even if it's not active, maybe just changes in the biochemistry of the plant because it's stressed with herbivory. Maybe that changes which fungi can colonize and be successful and which don't. So, yeah, definitely more to unpack with that.
I have a follow-up question on that actually. So if a plant that's being eaten has a more helpful arbuscular mycorrhizal community, have you ever tried taking that community and then putting it onto a non-eaten plant to see if it stays?
No, but that's what I'm going to do now.
Because we're always looking for these beneficial ones. I mean, with ectos as well, it's like, how do you choose the most beneficial and how do you keep it around? So be curious.
Yeah, I think that'd be you mean like a sort of almost like a plant soil feedback, I suppose, but
Mhm.
very targeted. Yeah.
Yeah, no, cool. Question from Tom May. Super to hear about your experiments. Can you comment on the way that the research you are doing could influence the investigation of traits? For instance, you mentioned phosphorus delivery. Are there specific traits that you suggest could be a focus based on your hypotheses to explain your observations?
Um, there could be, and I think yes, with AM fungi, there's this idea that certain families, like the Glomeraceae, are more associated with defense. That understanding, I think, is based a lot on root pathogens rather than other things. But and I think a lot of that is stemmed in the trait of being a high root colonizer, investing more of fungal biomass in the root rather than fungal biomass in the soil. And so I don't know, maybe it's intensifying competition for space with other things. Whereas those that are maybe not such heavy colonizers of the root, but more extensive exploration of the soil are maybe better at nutrient foraging or more efficient at nutrient foraging, but not so much at protection. So that's like a paradigm, I suppose, that people cling to, and I suppose I also cling to because there's very few things to cling to on this at the moment. But there's definitely things that need to be followed up from, I suppose, a traits point of view, from an ecologist point of view as well. There are consistencies with certain AM fungal taxa that show greater defense priming. You see it in the literature that certain species do tend to increase jasmonic acid, both with transcriptomic and metabolomic studies. They consistently show this, but I haven't seen anything that's then tried to link that to the fungus itself in terms of its biology or its life history strategy. But I think that's ripe for the picking. Connecting the traits to defense function, I think, would be awesome.
Cool. A follow-up question from Haggai. Um, did you look at diversity at the single pot level or at the entire treatment group? And if
Uh,
were they similar?
oh, to compare them. Yes. So the diversity was, so these were in the roots. So each sample was the root system of a plant, and there was a one plant per pot. Did I look to see the difference between each individual replicate of the treatment and the treatment mean? No.
Fun follow-up things to do.
Yes, I think it's so much more to do with the data, right? You can look at it. But yeah, that's very cool. Very good suggestion.
Um, Camille says, great talk, Adam. Did you or do you think that more diverse plant communities can also favor AMF diversity below ground that could in turn enhance plant response to stress?
Yes, I think that sounds reasonable. But I also think it probably isn't a rule because, at least from what I've seen, at least from a defense perspective, and also maybe other plant success parameter perspective, diversity can mean very little, uh, while composition, like who the identity of the fungi is more important in terms of the benefit. So if we're thinking of like stress resistance or resilience, um, diversity, I suppose, might provide more options in terms of function that's available within like the broader pool of species available to the plants. Um, but I suppose, yeah, it's hard to predict how that sort of plays out for individual species. So I think it'd be variable, and it would depend on which plants are within that community as well, in terms of if they're reliant, how dependent are they on AM fungi and their defense strategies.
Right. A question from Finn Bullman. Hi, Adam. Thanks for the presentation. In the experiments mentioned, did you have difficulty with AMF contamination in your non-AMF control plants? And I assume the follow-up to that would be, how do you avoid that?
Mhm. No, I did not have any contamination in this, but that does not mean it has not happened. So, so like in other instances, I think avoiding it is it's just about being careful. Where I have had some contamination, luckily it's always been in a limited number of reps, for example, or and where it has been, it's been very low. I suppose we always with AM fungi, the difficult thing is a lot of the work has to be done in pots, often with soil, or even if it is sterilized, irradiated, or autoclaved, or whatever you've chosen. Um, but I suppose the issue with AM fungi being so ubiquitous, like they're found everywhere, really, a lot of it just comes down to those small aspects of your experiment, I think, and just trying to be as careful that you don't have any contamination between treatments. I don't think there's any, I don't have any particular tricks other than just yeah, try and avoid and limit any sort of dispersal that could also be happening from outside, because there are, even though AM fungal spores are big, they still disperse through wind.
What is your favorite sterilization technique of to date?
I don't know if it's my favorite. In fact, it's not my favorite, but I know it's the best one would be the gamma radiation at 50K gray or above. which is really effective. It's just really logistically painful to organize. Anyone who's done it knows you just wish you could chuck it in the autoclave. But anyway.
Yeah. Well, thank you very much, Adam. It's been fantastic having you with us today. Thank you for answering our questions, and for such awesome work that you're doing in this area. So,
Ugh.
for everyone else, again, remember, same time, same place, next month. We look forward to having you. And also remember, upcoming later this year is ESA. So we will be partnering with ESA conference in December to have special symposia dedicated to mycological aspects. So we hope to see you there later this year. Think about registering and also becoming a member there. Thanks, everyone, and bye for today.